This brings us to our program for today. Today we have Professor Suraj Shankar from the physics department here. He grew up in Mumbai on the west coast of India. Throughout his childhood, he remained equally interested in science and in art, and the intersection of the two continues to permeate his work, as I'm sure we'll see today. He got his undergraduate degree in chemical engineering, decided to switch to physics for his PhD. He came to the University of Michigan Department of Physics as an LSA Collegiate Fellow and Assistant Professor in 2023 and has been here since. Please join me in welcoming Professor Suraj Shankar.
Thank you so much. Thank you so much, Christine, for the introduction, and thank you all for being here. It's wonderful to be here and to present and share some of the work that we've been up to in the context of the physics of active matter. So hopefully I will, through the lecture today, I will tell you what the words, at least in the title, mean. And hopefully along the way we'll have some nice pictures and videos to keep us engaged and awake in the morning. Okay, so where's my pointer? All right, so as Christine mentioned, I'm a physicist, but a lot of what I will tell you about actually has to do with the living world. And what I'm going to try to convince you today is why a physicist should and does study life and the living world, and what can a perspective of a physicist teach us about how the natural world and the living world organizes itself and how and what life itself is. And so let's start there. What is life? Life, of course, we're all familiar, we're all alive, and we have a sort of intuitive understanding of what living things are. Life manages to accomplish a wide variety of distinguished functions, and that we sort of quintessentially associate with living things. Growth, reproduction are most familiar. There's heredity and evolution. We have heredity of genes. There's also organization. We're all organized structures. We distinguish ourselves from our environment. We could constantly consume energy. We all had bagels and coffee and breakfast. We consume energy to eventually do work, including what I'm doing now, which is speaking to you. And we regulate ourselves. We regulate our body temperature. We regulate our internal body relative to the environment. And even though the environment gets warmer and colder, we maintain the same, right? And so living systems do all of these complex things, which are sort of distinguishing features of life, right? We think about rocks and salt and minerals don't do these things, right? So inanimate objects don't do this. And so all of these tasks, they come with a cost, right? And the cost is energy. We pay energy to do all of this, and we can estimate this. And if you start thinking about the energetic cost of being alive, this is starting potentially where physics starts to play in, and we can estimate how much energy does it take to be a human being. And if we go by the WHO standards of what the basic caloric intake is on a daily basis, that's the recommended number of kilocalories of food and nutrition we need. And basically, we end up consuming every day sort of a rate of a power of about 100 watts, which may not be that surprising because it's about the same power wattage of a bulb, of a light bulb. But if we compare this per unit mass of our typical weight to something as ginormous as the sun, something that's extremely, very, very huge source of energy, which is, of course, what's driving all of life on Earth. The sun pales in its sort of power output. And so it's just to emphasize to you how expensive it is, even on a per mass basis, on amount of how much we weigh, the amount of energy we consume to do the things that we need to do to stay alive, we consume a lot of energy, okay? And so one of the things I will try to tell you in the rest of this talk is where does this energy go and what are the consequences of this energy consumption into the remarkable and marvelous features that we observe in life, in the kinds of phenomena that living things display. And so this point that life itself was out of equilibrium. This is what physicists tend to call being out of equilibrium, because you're constantly consuming energy and you have a flux of energy flowing through you to be able to exist out of equilibrium, was recognized a long time ago by Erwin Schrödinger, who was famous, in fact, for his contributions in quantum mechanics. But he wrote this beautiful book. It's a very, very small book, and I recommend everyone to have a read. It's an easy sort of bedtime read, where he thought about what, the book is called What is Life, and he thought about what the physical constraints are of living things and what was needed, what the impetus was for the physical sciences to explain about the phenomenon of life itself. And he stated that how can the events in space and time, which take place within the spatial boundary of an organism, of a living organism, be accounted for by physics and chemistry, by the laws of physics and chemistry. And that's really the impetus for a physical scientist like me and many others and my colleagues to account for the phenomena that living systems display and explain it using the laws of physics. And similarly, perhaps less well known, but a remarkably pioneering developmental biologist, one of the first African-American developmental biologists to get a PhD in the US, Everett Just, even earlier recognized that the analysis of the state of being alive itself must never go below the order of organization which characterizes life, right? And so if you want to study the state of being alive, you have to study it at the level of the organization at which living systems are. You have to ask questions at the macro scale where living systems exist, and not everything must boil down to atoms and molecules. And that's part of what I will try to, again, emphasize through this lecture. And so the point I want to try to emphasize over here is that life is potentially, and maybe pose as a question, is potentially life should be viewed as something that emerges from the physical interactions and properties of matter, but it's a very special state, just like we have materials around us that are solids, liquids, gases. Perhaps life itself is yet another of these very, very special but emergent states of matter. And this view is really what in the now past 30 years or so has become codified into what's called active matter. So this is the term from the title of the talk. So what is active matter? Active matter refers to classes of systems that are made up of constituents, units, agents, what have you, not made up of atoms, but units and agents that consume energy. Right? So they consume energy on the small scale, and then you have many, many of these units together that they interact, and then they display remarkable organization on large scales. So that is a definition, if you'd like, of what active matter is. Now, it's a very, it's something that we've all encountered. Active matter, most famously, is on the larger scales, is what we experience and see in large gatherings of animals. Bird flocks, fish schools, they are examples of active matter on the very largest scale. But you can also go down on much smaller scales, and I will try to show you examples later on, including in some demos, that single cells, when they can aggregate in large colonies, can display collective organization, even objects that make up cells. So various molecules that sit within cells can all be fruitfully thought of as active matter. And in all of these cases, For instance, in the birds, of course, now each bird is able to fly because it's eaten food, and it manages, along with its neighboring friends, manages to aggregate in large collections to flock in remarkable patterns. Now, accompanying these natural examples in the past several years, now past 2 decades or so, there have been several ingenious realizations of such active systems in synthetic, completely non-living units, including robotic materials, grains such as rice that are vibrated on a plate, and also plastic beads that are motorized by chemical reactions. So you can have extremely synthetic, artificial systems that, if you are clever enough about how you drive them and how you pump energy into them, and we will see some examples, they mimic, and they are surprisingly well, they mimic features that we would think of and as distinguished features of living systems, such as collective motion, collective organization, and all sorts of pattern formation. Okay? And so this is the realm of active matter, and it's this view of thinking about living systems as an emergent state of matter, and once you take that perspective, then you recognize that there are many other, potentially even synthetic systems that can display the kinds of interesting, unusual, and special properties of life, but using ingredients that are non-living as well. Okay. It would be remiss for me to talk about active matter without talking about its most eponymous example, as I mentioned, the flocking of birds, which, after all, is in fact historically where the field started. So here's a beautiful murmuration of European starlings, and maybe I'll just wait for 10 seconds or so we can all admire what they're doing. Each of those black, tiny dot-like things is a single bird, right? And you have a huge collection of them, several, maybe tens of thousands, if not 100,000 of them, that are spontaneously, they're aggregated, they're flowing as if they were, if you squint your eyes and you blur things out, it sort of looks like a big drop that's sloshing out, freely floating in the sky. And they're doing this with no leader, right? So each of them is flying, they are interacting with their neighbors, and they manage to create, with no architect, with no choreographer, absolutely remarkable patterns on huge scales, and they do this by themselves, right? And so one of the puzzles that physicists looking at this had was how do they do this? How do they manage to coordinate their movements across tens of thousands of birds to be able to display this sort of remarkable behavior, right? And that's a film that I took off the internet. Okay, so this is motivated by questions of this is where the field really started. And in fact, it wasn't physicists who initially jumped in, but in computer scientists, because there was a lot of interest, in particular in the animation industry, to ask how can you mimic this on a computer? What do you need? What are the minimal rules you need to describe and display such collective motion, such flocking behavior? And Craig Reynolds, in 1987, came up with what he called the Boyd's model, where he had a simple caricature of birds flocking, nothing about the biology of birds, but just describing birds as particles which have a direction in which they want to fly, and they listen to their neighbors, and they try to line up with their neighbors, so everyone's going in the same direction, but they make mistakes. Okay? So very simple, relatively simple set of rules, but you put that on a computer, and voila, you get spontaneous collective flocking. with very, very simple rules. And these are the kinds of rules that went into simulations that, for instance, underlie the flocks of bats that you see in Batman Returns, and also later some of his work was in the context of the wildebeest stampedes in The Lion King, for which I think he got the Oscar as well, or the rare intersections where a scientist also got an Oscar. And so this was one of the motivations over here, completely synthetic, right? You don't care anything about the biology, you just want to mimic it on a computer and ask what minimal rules do you need to replicate the large-scale phenomena. And you realize immediately that very little is sufficient. And it was motivated by these kinds of studies that later on in 95, physicists took the same kinds of descriptions, but put on their physics hat, and recognized that this was a problem that material scientists and so-called what's called statistical physics, which is an entire field of physics that has to do with how interacting particles with minimal rules can display large-scale emergent behavior. And these guys, so particularly Tamas Vicek and collaborators, and then eventually John Toner and Yuhai too, recognized in 95 that flocking, taking this perspective of a material, thinking of flocks as a fluid that is spontaneously moving, was a useful picture. And these sort of minimal rules that Craig Reynolds had written down, wherein you have a bunch of particles, they all just move in some direction that they want to, randomly chosen, but they listen to their neighbors by aligning, and they make mistakes. So these ingredients, if you put it together on a computer, you get spontaneous collective motion of these arrows, these particles, which are all moving in different directions. They all decide spontaneously to move in some direction, and then you can study the physics of these systems. You can ask what the effective fluid dynamics of these materials are. And so that is the sort of brief history of the field. And more recently, in the past sort of 10 years or so, people have also figured out that you can make synthetic versions of these guys, of flocks. Here's an example of an experiment done by our collaborators, Denis Bartholo, and then a PhD student, Emily Chardac in Paris, where what they did is they took micron size, so these are tiny, very, very tiny, much smaller than the thickness of your hair, beads. They're plastic beads, and they have about 1 million of them in a thin channel. And if you activate them using an electric field that's out of the plane, out of the screen, then through some hydrodynamic introductions, they start propelling themselves. Each of the beads starts rolling and moving by itself, and when you have many, many of them, they interact and form these marvelous-looking flocks. Okay? Again, completely synthetic, but the rules, the minimal ingredients that I mentioned, which is you have particles that move and line up with their neighbors and make mistakes ever so often, you can emulate that on a computer. You can emulate that in plastic beads, right? With very different physics of how you realize it, but the large-scale properties and the large-scale collective behaviors are very reminiscent, and in some senses even not just qualitatively but also quantitatively similar to how flocking happens. Okay, so this is only to emphasize and point out that some features of for instance of a living system to spontaneously move and collectively move over here is recapitulated perhaps in completely dead things. Okay, so this was a bit of a preamble on the little bit of history of what active matter is, and in particular with regards to flocking. Now, one of the things that I mentioned, which is collective motion, all of these things are collectively moving. They're moving as in a coherent fashion, and they have spontaneous motion. So they're moving all by themselves. There's no other forces and fields that are applied that is telling each of those particles or each of those birds to move in any specific direction. They're all picking the direction to move randomly, and then they're coherently moving together through interactions. Okay? And so this, in physics language, breaks some fundamental symmetries of the problem. We are familiar with, we may all have encountered that in physics, symmetries are extremely important. They tell you about various constraints about the properties of a system. Now, the fact that active and living systems, the fact that they're alive, and for instance, they can display spontaneous directed motion, means that they break a very important symmetry, which is time reversal, which just means that if you reverse time, then the movie of the thing that you're seeing will look different, right? So the forward movie and the reverse movie don't look the same. That's what it means to be out of equilibrium. And if I take a walk this way, then the reverse of me walking will be me walking backwards, and those two actions are different, right? In equilibrium systems, if they are all at thermal equilibrium, such as dead particles, the actions that they exhibit, the behavior that they exhibit will be the same no matter if time is going ahead or backwards. Okay, so I have this, there's a very famous video from now, rather old, from 1950s, from David Rogers' group at Vanderbilt, which shows what you will see is a white blood cell, a neutrophil, which is going to be chasing this black dot, which is a bacterium, okay? And I guess I might have already given away the answer, but I have two videos here, and I'm going to play them, and I want people to raise their hands to guess which is the forward movie and which is the reverse movie. Okay, how many of y'all guess that the right-hand side is the reverse? And the left-hand side is the reverse? Okay, there's way more people who've raised hands saying that this one is the reverse. And that is correct. What you see over here is, in the beginning, the cell spits out the bacterium, and now the bacteria is chasing the white blood cell in reverse, whereas over here, the white blood cell is chasing the bacterium and then will now soon engulf and kill the bacterium. And so, while this is a microscopic example, I'm sure we've all encountered and seen, run and chase is a very common, as you're seeing over here. You have running of one organism and chasing by another. It's a very common feature, again, of living systems. Prey-predator interactions are of this kind as well. And this has a very distinct feature that if you run the system in reverse, if you run the movie backwards, it's a different, the movie is different from the forward run, right? And so these are examples, necessarily over here, of breaking what's called time reversal, because the reversed movie, reversed in time, is not the same as the forward movie. Okay? And so that was a biological example, but now we will look at a demo. Oh yes, good. We will look at a demo where, let's hope this works, where we will try to show you how surprisingly little, and in this case actually with rather mundane ingredients. So we're going to use ingredients which are essentially off the shelf. You can buy it. And we will use drops. We'll use drops of water mixed with a chemical called propylene glycol. It's a viscous, non-volatile substance. And then we have some food dye added to this liquid so that you can see it and visualize it. And what my colleague here is just doing is just cleaning up the glass so that the drops don't stick to it. And what we will show is that if you just place a bunch of these drops, then they too can, again, there's no biology here now, but these simple drops will manage to chase each other. and they start chasing, running and chasing. The green is chasing the red, and you see that. And once in a while they merge. There, that's a nice. Right? And over here, this is just water with some food dye, and they, there's nothing alive over here, but it remarkably manages to start exhibiting features that are very reminiscent of things that even single cells can do, right? And the reason a cell is able to do it is, of course, because it is consuming energy. Over here, this is actually relatively subtle. The reason these drops are actually able to perform this kind of behavior, active behavior, lifelike behavior, is actually because they're evaporating. And they're evaporating, and the evaporation is what drives, it serves as the engine for these drops to actually run and chase each other. And the moment they stop evaporating, they stop doing this. So all of this was just to emphasize that surprisingly simple deviations from equilibrium and very, very subtle, even subtle variations can actually allow non-living things completely, and sometimes very, very, very basic non-living things. This is just water and food dye and a little bit of alcohol that can now start, in appropriate conditions, can start mimicking rather complex behavior that we might associate with things like cells. Okay? All right, I'm going to switch back to the slides. Oops. Okay, all right. So this demo is actually based on a very nice paper and work from Manu Prakash's group at Stanford. Okay, and I encourage you to, you can even try this at home. It takes a little bit of finesse, but all the ingredients are things that you can get off the shelf. And so, talking about symmetries, one of the sort of overarching goals of this field has been asking, here are some of the questions that we as physicists are sort of interested in asking about these systems. And for instance, what are the unique consequences of being alive? Now that we have noted and seen that you can mimic life-like features in completely synthetic systems, what about life makes it unique? And what aspects of being out of equilibrium, being active of this kind, endows matter with these kinds of properties, with these kinds of lifelike properties? Are there general principles that we can understand and describe of organization, of self-organization? And can be, from a material science and from an engineering perspective, this is interesting because now we can ask, how can we emulate these properties of living systems, cells, tissues that do the remarkable things that they do in physical materials? Can we make and design materials that can heal by themselves, that can move by themselves? And then there's definitely all sorts of open questions about how should we understand biology. Is this a framework for understanding the remarkable things in biology? And as I mentioned already, we think as physicists, one organizing framework is to think about symmetries. I mentioned one very key symmetry, which is the reversal under time. There are also spatial symmetries that we care about, in particular whether systems are rotating, so they are so-called, they have a handedness, they're so-called chiral, or if they have orientations, if they have directional features. And that's what I want to spend the rest of this lecture telling you about. And then finally, you can also ask the kinds of questions that we're familiar with from everyone would have encountered. We learn in school, we learn about phases of matter, we learn about gases, liquids, solids, and so on and so forth. The same way, living systems, given that I already primed you in thinking about life as a state of matter, as an emergent state of matter, can we ask the same questions? Can we characterize, categorize, and understand how living systems also display distinct categories, distinct phases, and what are their properties? And can we use that as an organization organizational principle for understanding the diversity of phenomena that life displays. And so I want to spend some time, the next sort of half of this talk, I will spend some time talking about a class of systems that we've worked on for now several years, which goes by the name of nematics. And I will explain what a nematic is, and I will explain what I will eventually describe as an active nematic. But before that, maybe we'll look at an example. And so I'm going to ask one of my students, Maggie, to come and show you what, in this case, bacteria. So we're going to look at a swarm. So a swarm is, so that video, the video on the sides, which I will play in a second, is just a video of the same bacterium that we took previously. And Maggie will show you a real-life, real-time demo of what these bacteria, they're grown on effectively a thin layer of jello, agar, and if you give them enough food and nutrients and water, then they're happy, and these guys, they have tails that are what are called flagella that they beat to move fluid around and allowing them to swim. And if you have many of them, then they start swarming. They start flocking and swarming and collectively moving. Oops. Why is this not running? Okay. So on the sides, you will see a version of this particular bacterium that you can see the individual cells. They're rod-like cells that are swimming around. And in the middle, hopefully you should be able to see motion of these bacterium. This is a slightly lesser resolution than on the sides, but you see coordinated motion wherein you have all of these bacterium that are using their flagella to beat and beating their flagella to push water and use that to swim. And if you have many of them, then they form cohesive aggregates and flocks that allows them to move in a collective fashion. Okay? Are there, I guess we lost the previous spot which had more motion.
Yeah, I can look around a little bit.
Okay. Maybe we spend a little bit more time. This is always a bit finicky because, well, this is real science. Yeah, so you can see a little bit. You can see some of these bacteria sort of moving, and if you could resolve the individual cells, that's kind of what they would look like. And over here, yeah, you can see some single cells over here. Yeah, yeah, they're dots, and they're swirling around, and when they're dense enough, then they display these collective swirling dynamics. And again, this is completely spontaneous, right? All they're doing is they've eaten up some food, they're having nutrients, some sugars, and they're using their flagella, these filamentous appendages that they have, to beat them and then swim. And when you have many of them, then again, they spontaneously create these swarms, not unlike the kinds of bird flocks that I already showed you. But these are different because now they are on a much, much smaller scale. Okay, yeah, you can see them nicely over here. Fantastic. Thank you so much, Maggie. Okay, so I told you that I'm going to tell you what nematics are. And so this system is a kind of active nematic. It has, and let's go to what a nematic is. So a nematic system is, again, something we all have in our pockets. A nematic system is a material in which you have a bunch of molecules or units that are typically long. So in the molecular context, these are long molecules, which when you have many of them, they tend to line up. If If you have a bunch of long things and you put lots of them together, they tend to line up. And the material that you eventually get when you have a bunch of aligned long objects is what's called a nematic liquid or a nematic liquid crystal. And that is what sits in all of our liquid crystal display screens, in all our phones, in our monitors, in our computers, and so on and so forth. Right? So it's a technologically incredibly important material in terms of to understand its physics. And these are, as I mentioned, so it's a state of matter that is described by orientationally aligned long objects. And the bacteria that I showed you, or that Maggie showed you really, is yet another example of a nematic, because each of the bacterium was a long elongated object, and you had many of them, so that locally they all tried to line up and created similar kinds of order. They were all nematically aligned. And there's a very simple demo, this even I can do, which is just a plate with a bunch of rods, a bunch of toothpicks. They're all long, elongated objects. And, okay, good, so the camera is up. And all I'm going to do is agitate it. And so, and hopefully what you should start seeing is these guys are going to stop jumping out of the plate, but at some point they will, as you can only see near the edges, and even in the middle, they're starting to line up. Right? So there's no interactions over here. All they do, they're not attracting each other. It's just agitation and the fact that they're geometrically very long. They have aspect ratios, which are very slender, and immediately you see there's patches of aligned patches, which are pointing all in different directions. So you manage to get order of over here, which is orientational order. Everyone's pointing in the same direction spontaneously. All I did was randomly agitate it. Okay? So the same kind of physics of, this is essentially a point of packing long objects. This applies not just to toothpicks, but also to molecules and to bacteria, right? And so, and that's part of the reason why a lot of us in the field have been very excited about nematic systems, is because they're ubiquitous. Having long objects and having them dense enough is a very common feature. Cells oftentimes are very long, and as we've seen, things like bacteria are long, and they often occur in dense collections. And then when you have many of them together, and now if each of them has the capacity to move, so they've eaten up some food and they are able to propel themselves, they're able to swim, they're able to fly, they're able to crawl, then what you get is what's called an active nematic. So these are collections like the bacteria. This is another example. This is a different example of what's an active nematic. The dense collections of elongated objects. In this case, this video is from Zvone Medojic's group at Santa Barbara. What you're seeing in white lines, these filamentous objects, are really polymers. So they're long filamentous objects, molecules that are actually taken out of cells. These are particular kinds of polymers called microtubules that give your cell some integrity. They are like the skeleton of your cell. And they are now driven. Each of these polymers is driven by a motor, a molecular motor that really is like a nanomachine that takes in chemical fuel and walks on these filaments, causing the filaments to slide relative to each other. So you have a bunch of these filaments all assembled together in a two-dimensional layer, in a thin layer, and you give it chemical fuel, you add these motors, and voila, this fluid stirs itself. All by itself. There's no external pumps, there's no forces, no one's telling the fluid to go from one way to the other, there's no pressure, nothing. All this is a dish with a thin layer of this fluid, and you apply chemical fuel everywhere, and the whole fluid stirs itself, all by itself, right? And this is the kind of dynamics that you also saw in the bacteria. They spontaneously can stir themselves up and mix themselves up, and several people, there's some references here for the scientists in the audience who are interested, have been interested in asking what are the general properties. As I mentioned, having these sort of nematic systems is ubiquitous. You see lots of systems. I'll show you some more examples of where you see this kind of behavior. They're all over the place. And so rest of this talk, what I want to tell you is what we understand about these systems and how understanding toy systems and experimental versions and simulated versions of these systems is starting to help us understand some problems also in biology. Okay, so one of the key features of this active pneumatic, is that this is a fluid that is unstable. So if you prepare all of the material with all of these filaments pointing in one direction and you let it go, then what you find is that the whole material buckles. It sort of buckles in the plane, and this is because of an intimate interplay between the order on which direction you're pointing and the flows you generate. And an easy way of thinking about this is if you have a line of these rods, which are all, in this case, sliding relative to each other and pushing on their neighbors, and you poke it a little bit, then they're all going to start rotating because of the flows that they generate, and then you get these sort of remarkable patterns which come about, again, spontaneously. And when you wait long enough, eventually it gets this swirling dynamics that I already showed you on the previous slide. So we can also, that was an experiment, we can also replicate this on a computer. There's models, I will not show you the details of them, but you can replicate this sort of behavior, wherein over here I'm showing you the flow velocity. The video is looping, that's why it keeps resetting. That's the flow velocity, and over here each of these lines tells you the local direction in which you're aligned, you're pointing. And one of the things I want you to note is these green and pink magenta dots, which I've marked out. They are special points, which I will spend some time about. They are special points at which order breaks down. And these are what are called topological defects. And actually, if I can switch back to this for a second. Yes. I want to show an example. Okay, so this was our plate with toothpicks, right, that I managed to align. And there is a point that happened by itself, right, which I want to, there. Okay, there, right, so you see, let me just keep it down. That's good, okay, this is, all right, here. So that point, so you have a bunch of lines that are pointing this way, you have a bunch of lines that are pointing this way, and then there's a single point over here where you have no rods, and the direction in which everyone's pointing is, all the toothpicks are pointing in all directions at one point. You have a breakdown, it's a singularity of order, of the orientation, and this structure, where on one side you're pointing horizontal, on the other side you're sort of pointing 90 degrees to it. That kind of structure is called a topological defect. They are special singularities. Let's switch back to the slides. Thank you. So these are special singularities, well known, we have them on us as well. We can see them at the tips of our fingerprints, so I will encourage you to see it when it's brighter. Everyone has fingerprints which are formed of ridges, and these ridges are basically lines, right? And so they often have what are called whorls, which are comet-shaped. They look a little bit like this, like a comet, and then you have these threefold sort of Benz symbol-looking things, and in mathematics and physics, these guys are given names. This one's called a plus half defect, and that one's called a minus half defect. The reason they're called plus and minus half is because if I take the orientation in which the lines are pointing and go around in a circle about the center, the line rotates by 180 degrees, half a circle, when I go a full circle around, and it does this in the same direction, in the clockwise direction. This guy does the same thing, but in the opposite direction, so in the negative, in the anti-clockwise direction. So it rotates opposite to the direction of the circle, and that's why they're called a plus half and a minus half defect. Now, these defects, these singularities, they're special. They're very difficult to remove. Once they form, they're very, very difficult to remove. And that's what it means to say that they're topological. They're very robust to deformations, to smooth changes. And no matter what you do, they'll always be present. And they are sort of fingerprints of broken symmetry. They tell you what the system wants to do. They tell you that the system wants to line up, and in isolated points, you have breakdown of this order. And as we see in these active nematic films, they spontaneously emerge, they get spontaneously created out of the uniform state, and I'll let that play again, and what you see is that the plus half comet-shaped defect, it runs. It swims all by itself, and it starts flowing. And in a sense, you can think of these as sort of minimal particles that are effectively stirring up the fluid, wherein one of these defects is motile. It propels itself because of flows and stresses and forces that it's able to generate because of the fuel, the chemical fuel that was supplied to this material. And these defects have been of much interest because they offer a simple, they're point-like objects in this continuous fluid. So they offer a simple way to think about the effective dynamics and the effective properties of these systems. And they're well known from liquid crystals. They're also used in displays in molecular systems, but here we see them in these active systems where now they have spontaneous dynamics and they drive flows and they drive reorganization of the material. And they're present even in the bacterial case. They also emerge spontaneously in these films of polymers and molecular motors. And more recently, one of the reasons why these defects have been extremely exciting in the field and the community has been because people have recognized that these points, these singular points, these special points of breakdown of order are special not just from the physical and mathematical point of view, but also potentially biologically. So there have been a number of different examples in cells that you can grow in culture. So these are long, elongated cells that will eventually become neurons. And if you culture them and you grow them, because again, they're a bunch of long things, when they're dense enough, they form nematic patterns, and they can also form defects, which you see over here. That's a plus half with a comet shape. Here's the Benz-like, threefold trefoil-looking defect. And what you find is that the cells tend to accumulate. They form a mound and they accumulate at the positive defect relative to the negative one. And that eventually leads to further changes in the cell properties. In bacteria, this is a different bacterium, a soil-dwelling bacterium called Myxococcus xanthus. These defects are also formed. Again, these bacteria are long and elongated, and they move back and forth. And when you have many of them together, they form an active nematic. And where these defects are, you form multiple layers, which eventually is responsible and helps this bacterium survive starvation. And so these defects happen to be loci where you aggregate and then you save your resources and then eventually wait out. You form structures which helps you spore and wait out harsh conditions. And finally, here's an entire organism. These were multicellular experiments, but here's of bacteria or cell culture. But here's an entire organism, multicellular organism, what's called Hydra. It's a freshwater organism that is famous for its regenerative capabilities. You can chop this organism up however many ways you want, and then let it be until reaggregate and form the entire organism all by itself. These guys have, as you can see very clearly by eye, they have patterns of cellular structures that are lines that extend all across the organism, and they also form defects. These are nematic-like lines, and they form defects, and it's at these defects where you find the initiation of new tentacles. And this is a kind of sea anemone, so it forms tentacles. It also has a mouth and a foot, and these defects end up being locations where the mouth forms, the tentacles form, and the foot forms. So again, suggesting now in an entire organism that the defects have a potentially extremely important functional role for biology in deciding and dictating where important organs get placed, right? So for these reasons, the community has been very excited about studying both active pneumatics and these structures and these defects, because now it suggests that that's an organizing principle. And I'm not saying it's the only organizing principle, but at least in these examples, it suggests that there may be an organizing principle for thinking about how various complicated biological structures may organize and decide and take complex decisions about, for instance, where to form mounds, where to form layers, and where to form tentacles, based on potentially physical and mathematical principles. And on the counterpoint, not just in thinking about biology, from an engineering point of view, also there's been a lot of interest in asking how can we manipulate these defects and how can we play with them in synthetic systems. So here's a beautiful set of experiments from Caltech, where they take, again, a suspension of the same ingredients that I showed you before with these long polymer filaments that are taken from cells and molecular motors. But now what they do is they make the motors so that only when you shine light they become active and they crawl and walk and they consume energy. When you don't shine light, they're all floating around freely. And what you can then do is now ask an engineering or design question, which is you have now this material, you have this fluid which is capable of forming these self-organized structures, these pneumatic patterns, these defects, whatnot. And you can use light. You shine light in different locations. You self-assemble spontaneously structures like these defects. Now these are aster-like defects. So they're not dissimilar to cowlicks that we have on our heads. And now you can move them around and you can play with them as you will, right? And in principle, this now begs the design and engineering question of how can we take these ideas of, just like biology is potentially using these defects to organize and decide how to pattern tissues and cells in different locations, can we do the same thing in synthetic materials to pattern and design them to do complex functions and complex tasks? And so here's a simulation of a slightly different kind of manipulation of defects, wherein we are patterning the forces and stresses by this activity, and we cause different kinds of defects in this pneumatic to now braid around each other as if you were braiding hair. And so this was actually work done with a very talented then undergrad, now grad student at Chicago, and other collaborators at Santa Barbara. And so we're beginning to, we and many others, there's other people as well who are thinking about these things, are beginning to start to understand the rules by which we can not just understand the dynamics and mechanics of these patterns and these defects, but also start controlling them, and controlling them potentially as a way of understanding how biology may do it, but also understanding and trying to get down the rules for how we should do it to design new materials with new and interesting properties. And so much of what I told you and what I have been telling you has been in the context of fluids. More recently, we have also been very excited about asking what happens when you think about the same kinds of structures, but in a solid. And why solid? Because if in many cases, many of these organizations that of cells and tissues that I showed you, they do move around, the cells move relative to each other, so it behaves like a fluid. But there are many instances where that's not true, that's not the case. Your tissues and your organs, they're all staying put. The cells in them are not always moving around. And so our skin is not flowing off of us. And so they're all solids. They have rigidity, they have some elasticity to maintain shape, right? And so we can ask, what are the consequences of the same kinds of principles and the same kinds of ideas in a solid material? And over here, and one of the points that I want to make over here is that these defects that I mentioned to you already, these topological defects, they are able to move in a fluid because of forces and stresses that they generate. But in a solid, they can't move, but those forces and stresses are still present. And so then one immediate potential way of relieving those stresses is by failing, by fracture. And so these were very beautiful experiments done by collaborators, Mike Morrell at Yale, and a very talented postdoc, Shankar, who's now moved on to Northeastern, where they assembled now a two-dimensional layer of, again, these are various other polymers and motor proteins extracted from cells. These are a different set of polymers and motor proteins. These are, in fact, the same things that make muscle. They're what allow muscle to work, actin and myosin. This is the machinery that allows you to contract muscle and perform move. But now, instead of having them organized in a cell in tissues, you can extract them and put them in a Petri dish. And if you make a thin layer of them, these guys, so like muscle, it's a solid, right? It's not moving about, it's not flowing. These guys also form a thin sheet of a solid, but when you provide chemical fuel, what happens is it tears itself apart. Right? And that's real time, so it tears itself apart very quickly. And so you have a situation where you have this thin elastic sheet which is just ripping itself apart, and if you pay attention, you see that there is these patterns which are all reminiscent and actually exactly what I already mentioned to you. They are topological defects. You have patterns that look like the plus half comet and the minus half trefoil, and they're all over the place. And if you, one of the things that we did is to analyze this and to point out that just like in the fluids where these plus half defects are able to move by themselves because of these forces that they're able to generate, in a solid, those same forces cause them to pull apart and tear the material. And here's a simulation sort of showing you what that looks like. And one of the things that we've been interested is now to start asking questions about healing, about self-healing and resilient materials. How can you design not just materials that flow at will and spontaneously and autonomously, but now materials that also break and heal as and when you demand, right? And this is something that biology does all the time, right? When you get a wound, you heal immediately, and at the same time also when you're developing, when you start out as a single cell and then you become become an embryo and then you become a macroscopic organism, you start out with all these cells that are proliferating and growing, and then they have to flow around to move to the right places to be the organs that they need to be. And once that's done, they need to stop moving so that you stay put and the organs remain where they're supposed to be. So cells and tissues constantly do this. They switch states of matter. They switch from being liquid-like to solid-like, and they're able to heal themselves. They're able to break when they need to. And question is, can we start learning from biology, and can we start learning from these synthetic, bio-inspired or biomimetic systems to learn the rules from a physics point of view, and then potentially start designing materials from an engineering point of view to do these tasks just the way as biology does it. And so with that, I will conclude. And that's not that recent picture of our group, a very still nascent group. And so the main takeaways I want for you to leave here with is that one is that life itself is more than just molecules. It is, and perhaps a fruitful way of thinking about it is to view it as an emergent state of matter, just like we view and understand other phases of matter. And that perspective immediately forces us to acknowledge the fact that life is sustained by energy input, which is activity. And activity and mechanics in these contexts, particularly in biology, are extremely crucial. And this perspective, from a physics point of view, offers a different route than the molecular view of understanding how collective behavior and collective organization in living systems occurs. And there's many open questions, and the field is very much still very young and very open. And so it's a very exciting time both for us and for students, and hopefully for all of you as well. So I will conclude there. Thank you again for being here.
right, thank you very much. We'll give just a few minutes. Those who need to leave can head out now, and we'll be setting up a microphone over here. All right, I think we're ready for our first question in the room here.
He's more interested in the scientific. Why was it needed? What did you remove the static? What is this?
Yeah.
Yeah. So we had a question on the equipment.
Oh, yeah.
So what is this device? This is removing the static.
It's not quite removing static. It's mostly just to clean. So you can either clean the glass on a flame. That's just a corona discharge to electrically clean off the surface. That's it. It's just so that the droplets don't get pinned by dust and random stuff. But you can just do it on a Bunsen burner, which is also what we did. But for that, you just have to wait for it to cool. That's the only difference.
Okay. Any questions?
Yeah.
Thank you.
All right, our next question.
All right, thank you for the lecture today. One of the topics I'm studying is just what is life, and I appreciate the reference to Schrodinger because arguably that led to molecular biology. But is there any do you think there's a consensus in the field now on how to define life? Is it even definable? Like, because that's kind of one of the issues with that. So I'm just curious, given kind of your connection of your work to that definition, if you had an opinion on that.
Yes, no, thank you that's a very, very deep and important question. I don't know, I feel like I don't have a good answer for is there a consensus, because I feel there are definitions. There's lots of people who think about the origin of life, including not just biologists, but material scientists, chemists, astrobiologists. There's lots of people who are interested in finding life on other planets. And there are working definitions. The main difficulty with defining what life is we have one example. That's it. We only have life on Earth. And that's the only one that we know that works. And then extrapolating from there is, we all know how difficult it is to extrapolate from one point. And that's really the difficulty. So we know of properties that life on Earth has. Now, whether that's all necessary, perhaps not. I don't know. And that becomes extremely important when we start thinking about especially searching for life on other planets, because there's no reason that the chemistry anywhere else has to be the same to have life the way it is over here. And perhaps games and stories of the kind that and explorations of the kind that I talked about and suggested point to a feature, which is that maybe some of these properties that may seem very lifelike might be obtainable with very, very minimal, very simple ingredients, even with very simple chemistry, and that may help refine or recognize that actually this boundary between life and non-life is actually much blurrier than we might imagine. And so
quick follow-up on
yes.
that point. Do you think viruses are alive?
Say it again?
Do you think viruses are alive?
Oh, that's difficult. I will not.
I thought you could help me out, maybe.
Yeah. Happy to chat more about that later, but I don't want to get called out by a virologist on that.
Hi, thank you for a wonderful talk. I had a couple questions, if I'm allowed. One is that I'm aware of you showed some light used to move or motivate particles, in effect. And I'm aware of a couple of therapeutic uses. I'm wondering if it's connected to that. One is the LED face mask that I think is supposed to treat wrinkles or something, I don't know. And the other is a handheld laser that's used on the scalp.
Yes.
Some kind of therapeutic use. Do you think those are based on any principle that's related at all to what you were showing?
Not quite. I don't quite know how the laser-based either hair removal or wrinkle removal mechanisms work. My suspicion is they usually work with heat and less so actual, because they usually have visible or infrared radiation. The examples I showed you where light was manipulating these molecules, the molecules were engineered to be sensitive to light. So there are certain molecules that actually light sensitive, including the ones in our eyes. And if you engineer some of them to attach them to these motors, then they become sensitive to light. Now, in terms of sort of broadly thinking about therapeutic opportunities, there are people who use some of these ideas, maybe not with light, but with other mechanisms, like magnetic fields, electric fields. And there's lots of interest, for instance, in devising and thinking about active agents for drug delivery. And ingestible micro-robots that sort of swarm and navigate complex, your gut to deliver and to deliver drugs to help cure, deliver chemotherapies to cancer tumors and things of that kind. So there's several people in bioengineering who think of this, the same kinds of pictures of thinking of active agents and active particles and controlling them oftentimes. Well, sometimes with light as well, but oftentimes heat and electrical or magnetic fields because they are much easier to use on human bodies and use them to control and for lots of therapeutic and diagnostic purposes. And so that's a burgeoning, new and burgeoning field as well. Hopefully that.
Okay, yeah. My other question
Yeah.
was, when I thought of first was, it makes sense that swarming birds would stay in formation for migration purposes, but a murmuration isn't going anywhere.
Yes.
So what do the birds benefit? Are they scooping up insects, or are they getting exercise, or what's the benefit?
That's fantastic. Thank you for that. So that's, again, we honestly don't quite know. The common thought and the expectation is that the birds in murmurations do benefit from being in these large collectives to help evade predators. So if you do see, and so in Rome, these European starlings, they often, I think in August or so, every year, actually even over here, earlier in the year when it was colder, crows would form huge flocks. I tried to get videos of them, but I never was quick enough, on top of the Biological Sciences building. They would form huge flocks. I have no idea why they do it. But for starlings, the thought is that they form these murmurations to help evade and escape predators, because if you're solitary, then it's much easier for a peregrine falcon to come and catch you. But if you're in a group, then you essentially sort of confuse, and you have too many signals for the falcon to try to follow, and too many other sort of praise, and then that way you sort of save the group rather than sacrifice the individual. And this is the thought even for other organisms. Usually it's predator avoidance or predator escape. There may be other migratory aspects for other organisms. It depends then very much on the organism at hand.
Of course, it takes energy to
Yes,
to
yes,
fly,
and so that's why the why is always a much harder question. That's a question of ethology or behavior and biology in general. The how is somewhere where physics can hopefully say an answer. That's why I focus only on the how.
Thank you.
Thank you. Oh, careful.
Hi, thanks for your great lecture. Are there any limits to the scale of this sort of process? Do we see it at, say, a geological or planetary scale?
Oh, that's fantastic. Yeah. So I would say no, there shouldn't be limits on this because the only ingredients I mentioned, most of the motivations for the field came, of course, from biology, and that's macro, and then eventually we went smaller. But there's no reason why that should be the only case. There are people now building macroscopic realizations, or even larger, so using robots and things of that kind. But the idea that you have systems that are constantly maintained with energy input, that's not surprising. That happens on every scale. Stars do it. You have planets are constantly bathed in sunlight and energy. The atmosphere, geological processes also have constant fluxes of energy. Now, the question is whether this perspective plays a particularly beneficial, is useful over the tools that people already use in these other fields. That I don't know. But I would like to hazard a guess that maybe. We just haven't looked enough. And maybe there are, even on planetary scales or, beyond, there may be systems that, if we think carefully about, then this may be a useful perspective to think about collective organization.
The system that comes to mind is weather, for me.
Yeah. No, absolutely. Weather. So, we have decent ways of understanding how weather works. They're not perfect. Whether this view of and the framework of active matter is more quantitatively useful, I don't know. That's partly just because no one's looked carefully enough. Actually, maybe I shouldn't say no one. We haven't looked. But these are the kinds of things, as I said, it feels very young, and there's lots of open questions and lots of open scales as well. So we're excited, and if you have thoughts or you know people who are interested in this, then that's fantastic.
Thank you very much.
Thank you so much.
Let's go to a question online now. What about the surface-to-surface contacts between these filaments, and also the role of rigidity of these filaments when they line up?
So surface-to-surface.
Surface-to-surface contacts and rigidity of the filaments when they're lining up.
In the videos that I showed, I'm guessing.
Presumably.
Okay. Yes. So the filaments, these filaments that formed the pneumatic in the videos, they're so-called microtubules. They're very stiff, but they do bend. You could see them sort of bend around, and so yes, the flexibility is important because if they're very rigid, if they're like toothpicks, then the quantitative features are different, but qualitatively they will still do toothpicks. And actually, people have experiments with grains, tiny metal rods, which you vibrate, and they display the same phenomena. Bacteria are much shorter, and they're not really flexible, and they also display the same phenomena. And so, flexibility is important, but it's not essential.
Okay, let's come back to the room.
Thank you for a fascinating lecture. I was curious, you mentioned that active matter is a relatively new field, but I kind of view it as like related to the study of complex systems, right? That has the kind of a long tradition in physics. And you mentioned that you need to integrate over scales. There are tools like the renormalization group that help us do that, right? So what kind of distinguishes, what have we learned new kind of, what's the focus, I guess?
Yeah, so I'm not saying that, yes, of course, physics has, again, long history of thinking about complex nonlinear dynamics, weather was mentioned as well, and there's lots of people who've been thinking about modeling weather for a long time. Fluid dynamics is a much older field. And renormalization group, yes, also provides a schema for how to integrate over scales. The point is conceptualizing and putting a boundary and an umbrella around systems that are distinguished by the fact that energy input is local, not global, not on the outside, where you have like an external electric field or gravity that's telling everyone to go in one direction. Here, locally, each individual particle is able to make a choice, cognitively or otherwise, to do something by itself. Right? That umbrella, recognizing that was a distinct enough class of systems worth studying in itself because there were common principles there, that's much newer. There are ideas about that go even a little bit further back, but really convincing physicists that this is an interesting thing and using the tools of physics. And actually in 95, for instance, the reason all of this became possible was because of the advances, for instance, in the renormalization group that had just happened. And so we needed all of that, the skills and the tools that people had developed to be able to apply it to these newer systems.
Great lecture. And I just wanted
you.
to ask, does the murmuring effect in birds, does it only apply to birds, or could it also apply to a large crowd of people,
Yes.
say in concerts?
Yes.
Does that apply, same concept?
Absolutely. Fantastic. Thank you for asking that. Yes, there's some beautiful work done by a bunch of people, including by Denis Bartolo, who I mentioned, who had these experiments with plastic beads that flock. There's a bunch of people who study human crowd dynamics, and including in concerts, as you mentioned. So mosh pits are a fantastic example of collective swirling and swarming, where there's no organizer, there's no leader telling who to go where, but people spontaneously organize and form vortex-like structures and flows in in sort of large festival events where you have large gatherings of people. You also often see spontaneous oscillations, or if you're waiting before a marathon starts, you see waves that propagate. Again, you don't know any of this at the single individual level because you don't see it, but if you zoom out, you do see patterns that propagate across large collections of human beings. And again, the rules are similar. They in detail may differ, but the conceptual picture is very much the same. Yes. So human crowds are also very much active matter, and they also display all sorts of collective organization with very, very simple interactions and very simple rules. And this becomes extremely important when we start thinking about what are the ways in which you want to mitigate, for instance, disasters and disastrous stampedes, right? So if you want, in terms of human crowd management, if you have large collections of people that are leaving a concert, leaving an arena, you want to ask, and it's from a design question also, how should you design your space so that people are able to now navigate and move with the least amount of issues and problems and that least amount of damage. And you want to, and so there are a large number of people who are thinking about human crowd dynamics also from this collective macroscopic level, with a lot of interest in thinking about how crowds behave in dense environments, including at festivals and concerts and marathons.
Thank you. One more question.
Yeah.
Does the active matter, can it be attributed, as we've seen with the flagellated bacteria, like microscopic organisms bonding together and just forming an active matter, can it be attributed to larger organisms forming and being more organized in a sense, as in a human body is a lot more organized than, and then there's a lot more microorganisms that are responsible for the organization. Can that active matter aspect of it be attributed to the organization that eventually came to be emerged?
Potentially. I'm not entirely sure I followed. Do you mean, are you asking about whether the ideas of sort of the collective behavior of individual microscopic organisms are the same ideas also applicable to asking about multicellular organisms? Is that what you're asking?
Yes. So, yes, that is what I'm asking. As in the idea that single-celled organisms band together to form an organized entity.
Yes.
Can it be attributed to larger or multi-celled organisms?
Yeah, no, absolutely. That's another very, very deep and important question about the origins of multicellularity. So we are all multicellular organisms. Microbes are unicellular, they're single cells, and there are a lot of questions about as you correctly mentioned, that when individual organisms, such as microbes, they display collective behavior, they're coordinating with their neighbors, they're coordinating with everyone else. And at some point in our long history of evolution, that same kind of not the precise one, but similar kinds of collective behavior has and was harnessed by other kinds of single-celled organisms to then recognize that it actually helps to be together, to be stuck together, and give the onset for the very first multicellular organisms. And this is yet another marvelously fascinating field that there are several people who study the emergence and origin of multicellular life and are thinking about it also from this perspective of when does it benefit to be a collective? When does it benefit for individuals to band together and make a unit, which then undergoes biological selection. The same ideas also happen on much larger scales, where if you think about insects and social insects, things like ants and termites and bees, they're individuals, again, they coordinate their motion and they coordinate their behavior to display emergent and remarkable kinds of outputs, everything from constructing huge mounds and nests to being able to forage and to have agriculture, to be able to raise a brood and have social hierarchies all within them. And these, and oftentimes social insects are called and viewed as superorganisms because the entity now is the colony. And they're all, although they're all individuals, the colony is the unit on which biological selection happens, and they all, again, benefit from displaying this sort of collective dynamics and collective behavior. And I think this is far more general than we might naively imagine and suspect. Humans, of course, have civilization, and we have social interactions, and we have collective interactions, which allows us to have society, but so do animals. And increasingly, we're recognizing that every animal, or most animals, have some version of this at different levels, and at levels that we may have not previously have been able to access or observe. And so yes, I do think that this sort of organization and hierarchy of thinking about collective interactions leading to collective behavior and its implications for what counts as the unit of biological selection, that's a recurring feature at every scale. Thank you.
Go to another question online. Can you explain the role of RNA as an emergent state of matter?
Okay, so RNA is, well, it's a molecule. It's I'm not, again, sure in the context of what it particularly means to be, for a molecule to be an emergent state of matter. Usually, states of matter refer to a property of many interacting objects, not of individual molecules, so I'm not quite sure what that reference is, but maybe they're asking about the RNA world, which is a hypothesis about the origin of life, having started with ribonucleic acid, which is RNA. I don't have much to say about that, but because that's way out of my field. It is an interesting set of concepts, but the origin of life is outside my...
Can you talk about RNA and DNA as active matter?
Okay. So they themselves, as polymers, so these are RNA and DNA are molecular carriers of heredity. They are what allow us, DNA at least, is the ingredient that carries our genes and is what, again, biological selection and evolution act on. By themselves, they aren't in any sense active, but in conjunction with, as they live inside cells, they are subject to processes that are active, and those processes are informational, wherein you have other molecules that have to go, bind to RNA and DNA, read off them, read the code, the genetic code that is there, and to do that you have to unfold this polymer that is wrapped up tightly inside your cells, which is a mechanical process. So you have to apply forces, you have to unfold it, read it, refold it, put it back, and so all of those processes are very much energy consuming and are active. And there are people who study the active matter of chromatin, which is a condensed form of DNA, and ask how the energy consumption that is present. The cell spends a lot of energy in using machinery to read and write and unpack and refold these complex polymers, and you have to do this correctly, right? And that's always the task, which is all of these processes with molecules. You are trying to make decisions on them, and biology wants these decisions to be very accurate, very precise, because you want to, make your limb in the right shape and the right place, and so on and so forth. But because you have fluctuations, you always make mistakes. And so another place, very important place where biology and cells spend a lot of energy is what's called error correction. So you proofread, like we do when you write something, you proofread, you go back, you spend more energy to read it again and correct your errors. Biology does exactly the same thing. And that's yet another world at the intersection of information and active matter in terms of understanding how the encoding and decoding of information in living systems requires constantly the churning of energy to make sure you make decisions accurately. So hopefully that's answers sort of tangentially in terms of what active matter may have to say about RNA and DNA.
Oh, you have a question? Or-
Yeah, I have a question.
Yes, thank you.
So it's about wounds and healing. So you said that there are systems that do this. So I wonder if you could give an example. And is that really what happens when, like, I have a scrape or a cut?
So when you get a cut, so your skin is what's called an epithelium. It's a single layer of cells, and there's many other things underneath. When you typically get a cut, there's many things that happen. One is you have an immune response, where your immune system recognizes there's a breach, and then you get inflammation and so on and so forth. Your blood clots to stop bleeding. Then what happens is your tissue, through a variety of means, both chemical and electrical as well, manages to sense that there is an opening, and the cells at the wound, at the edge of the wound, they migrate, and they slowly crawl, and the rest of the tissue is solid-like, but the edge fluidizes. They manage to become fluid-like so they can fill up the gap, and then when they fill up the gap, then they stop moving, and then you've healed. And so people, you can recapitulate this in what's called a wound healing assay, because it looks exactly like what it sounds. You take a tissue and you cut it, and then you wait and you see how it heals. And people use this as a model to studying migration and how tissues especially switch from being solid-like to fluid-like transiently, and then revert back. But in the actual body, there's many more things that happen because they also couple to vasculature, couple to immune response and whatnot. But was this what you were referring to, asking about? Okay, thank you.
One quick final question.
I was curious, what is like unique about the way that active matter breaks time symmetry? Because to me, most classical phenomenon break time symmetry. Like I was just thinking, like a ball rolling down a hill is not the same as the ball going back up.
Yes, thank you, Ella. That's a very subtle point. I was. It means that you paid attention. So yes, you're right. So the subtlety over here is the point on thermal equilibrium. So in thermal equilibrium, you're not just going to the ground state. You have some finite temperature, and so thermal fluctuations keep you at some fixed energy and on some average energy that your trajectories going one way are the same, the statistics and the probability of going one way are the same going in the backward direction. So it's not a statement of, yes, if you have just a ball rolling down a hill, and the reason there the ball stops when it goes to the valley is because also you've reduced your energy, and there's no other fluctuations to maintain the energy. So you've actually lost all the energy that you, if you have friction or something, then you've lost all the energy. Otherwise, you just, if you were conserving energy, then you would keep going up and you would oscillate, right? And so the only reason if you have a ball rolling down a hill that it stops at the valley is because there's some dissipation, there's some friction that takes away that energy and it's lost forever, right? But at thermal equilibrium, that dissipation is balanced by kicks, random kicks that temperature gives. In active matter, that balance is broken. And however subtle of a breakdown of that balance is sufficient to give rise to all of the complexity that we see in active systems.
Thank you.
Okay?
All right, we're at the end of our time. Let's thank Professor Suraj Shankar again.
Thank you.